Hello everyone, welcome to Exam Essentials. Today we will go through some of the most frequently and commonly asked questions of Biology Paper 2 of Edexcel syllabus. So in this video we will be covering up to last 5 years of questions, so I would suggest you to not skip anything and go through the whole video so you can ace your exams. So without wasting any more time, let's jump right into it. First off, we have describe how nephron absorbs glucose back into the blood from the filtrate. So we know in nephron, glucose is absorbed back into the blood by selective reabsorption, which involves the process active transport, and active transport needs energy from respiration, and all of this occurs in the proximal convoluted tubule of nephron. So this is the answer. Glucose is selectively reabsorbed by active transport using energy from respiration against a concentration gradient at proximal convoluted tubule. Next off, we have hair color in cattle is controlled by one gene. So this is the information. Then they've asked us state what is meant by the term gene. So gene is just a part or section of DNA that codes for protein. So for 1 mark, this is a section of molecule of a DNA that codes for a particular protein. A student uses this equipment to measure the energy content of transgenic soybeans with energy content of non-transgenic soybeans. Describe how the student could use this equipment to make a valid comparison of the energy content of two soybeans. So this is the method. We are burning the soybean and measuring the temperature increase, with which we will calculate the energy released or the energy of the soybean. So they've asked us to describe how the student could use these equipments. So first off, we will use the mounted needle to burn the soybean under the test tube using a Bunsen flame. Then before that, we will have about 20 centimeter cube of water into the boiling tube so that it can absorb the temperature, and place a thermometer so it can measure the temperature rise. So this will be our answer. Add 10, you can add 10 or 20, either one is fine. 10 centimeter cube of water to the boiling tube. Measure the mass of soybean and mount it on the needle. Then ignite the bean on fire and hold it directly under the boiling tube. Igniting the soybean again and again is necessary until it is burnt completely. Measure the rise in temperature using the thermometer. We should also repeat it 10 times for reliability. Next off, we have a calorimeter. So the diagram shows different type of apparatus that can be used for investigation. Explain 2 reasons why this apparatus gives a more accurate value than the apparatus used by the student. So this apparatus has a lot more of features than the previous one, such as the stirrer. So stirrer is generally used in calorimeter to distribute the temperature or heat more evenly. Then there is oxygen gas inlet. So oxygen is needed for combustion or for complete combustion. So the oxygen inlet ensures the complete combustion of the food sample. Then we have insulated outer container. So from the name we can guess that It is insulated. That means it will prevent the heat loss from to the surrounding. And we have steel container with the volume of water or the container directly in contact. So steel, we know steel is a good conductor of heat. So as it is contained in a steel container, the heat will be quickly transferred to the water. So all of this does what? All of this gives a more accurate value of the energy content because less heat is lost due to all of these features. So we had to explain two reasons why this apparatus gives a more accurate value. So as I've discussed, NE2 is fine, oxygen inlet, which ensures the complete combustion, insulated coat, and ignition coil in equipment reduces heat loss to the surrounding sphere to provide more even temperature, and the sample is placed in steel so steel can transfer heat to the water. Loss of rainforest causes an increase in atmospheric carbon dioxide. Explain why negative effects of an increase in atmosphere. Explain the negative effects of an increase in atmospheric carbon dioxide. So we know carbon dioxide is a greenhouse gas. So it causes global warming, and global warming can cause the melting of ice caps, climate change, and many more. So, carbon dioxide is a greenhouse gas which can trap radiation from the sun and cause global warming. So, this also causes the melting of polar ice caps and climate change. So, this can further affect the food chains or migrations, ocean acidification, and many more. Now we have question about vaccination. So they've said that immune system of dogs works in similar way to the immune system of humans. Explain how rabies vaccine can prevent dogs from developing rabies. So in vaccination, we inject a person or the animal or the subject with weak or inactive or a pathogen with the same antigen as the disease causing pathogen, but it is not active such that such kind of pathogen will be injected into the subject. This will be detected by our immune system, so they will alert the white blood cells, especially lymphocytes, so they will start making antibodies. So, but in reality, the pathogen that we have injected, they do not cause the disease, but they just have the antigen, and our lymphocytes did what they already made the antibodies, which remain as memory cells. So when the actual pathogens such as rabies, the actual rabies enter the body, then they can produce the antibodies much faster and in more quantity. So this is what I've written. Vaccination includes injection of these pathogens whose antigens can be recognized by lymphocytes, which produces antibodies specific to it and remain in blood as memory cells. When normal pathogen enters, a secondary immune response is triggered where more antibodies are produced sooner. A new RNA vaccine is being developed to treat rabies. RNA that codes for parts of rabies virus protein code are injected into the body cell.
Cells then take up the RNA and produce the viral proteins. So this is the important part. Should mark that they produce the viral proteins. Describe how the student could describe how the cells use the RNA to produce the viral proteins. So when we look it at once, it seems a very unfamiliar question. It's very unclear what they're asking. But if you break it down, that RNA is being used to make what? To make produce viral proteins. So, but the important part is to make proteins. And in our syllabus, we only know two steps how to make proteins in protein synthesis. One is transcription and one is translation. So here we will talk about mostly translation. That's what they are asking us to talk about. So it is a very layered question, but if we break it down to key points, we will soon discover what they're asking. So here, ribosome binds to the RNA, this RNA, and translation occurs. RNA molecule brings the amino acids to the ribosomes. Yes, RNA has the codons which code for amino acid, and the anticodon on the tRNA binds with the codon, producing a chain or amino acid chain, which further gets converted to protein. Describe the role of one named mineral in the plant growth. So there are many minerals. Of them, the most two or the important, most important two are nitrates and magnesium. So magnesium helps to make the amino acids, and our magnesium helps to make the chlorophyll for photosynthesis. So this is a straightforward answer, that magnesium is needed to make chlorophyll and nitrates are needed to make the amino acids. Describe how water is transported from the soil to the leaves in the plant. So from the soil, that is to mark. We know in the soil we have the roots, so the water must travel or be absorbed by the root hair cell. Then it will go up to the leaves. So for that it has to travel through the xylem vessel. So xylem vessel carries the water from the roots to the leaves, and it is with the help of transpiration pool. So that is our answer. Water moves by osmosis into the root hair cells from higher potential to lower potential. Then it is pulled up along the xylem vessel by the transpiration stream or transpiration pool due to water loss by evaporation from stomata in the leaves. Analysis of composition of urine can be used in the diagnosis of problems with kidney function. Urine samples are taken from 3 patients and results are shown. So this is the result. Patient W has glucose absent in their urine, water content normal, protein present. So this is not normal. That means if a person has protein in their urine, they have some fault or problem at their glomerulus, because this is where the ultrafiltration takes place to filter out the big particles or molecules like protein. So we have a problem at the glomerulus or in the process of ultrafiltration. Then comes patient X, who has glucose present in their urine, but other two are normal, or water content is normal, protein is absent. So their selective ultrafiltration is normal, but they have glucose present, which is also not normal. So why? Because glucose is generally reabsorbed back into the blood by active transport using energy. This happens at proximal convoluted tubule. So the selective reabsorption is not happening. So they have a fault at proximal convoluted tubule. So therefore the selective reabsorption is not happening properly. Then we have patient Y who has glucose absent but water content very high and protein absent. So this person have problem with the water content which is very high. That means they their hypothalamus gland or the pituitary gland is not producing enough ADH for the walls of collecting duct to be enough permeable to absorb the water, reabsorb the water into the blood. So this could be a problem, or they have a problem with the collecting duct or the walls of collecting duct because they're not permeable enough. That's why water is not being absorbed. So this is the answer, that they have fault in ultrafiltration, as I've said, in glomerulus, because protein is a large molecule which shouldn't pass through it. Then patient X, there may be fault at selective reabsorption, convoluted tubule, because this is where it is taken back into the blood. And patient Y, there is fault at reabsorption of water by osmosis in the collecting ducts, because it stays impermeable, which may be due to less release of ADH, or it could be a fault itself. Describe how to taste for glucose in a sample of urine. So it's very simple. We will use Benedict solution and warm it or heat it, and glucose, if glucose is present, it will turn brick red. Describe how the scientists could determine the number of each plant species per meter square in each field. So, in order to determine the number of any species or each species or anything in a field, we will use quadrant. So, using quadrant requires to use it or place it randomly in a field without any biasness. So we will place it randomly at random coordinates. Then after placing it randomly in different coordinates, we will count the number of species within the quadrant at each coordinate. Then we can find the mean, and we will divide that number of species by the area to find each plant species per meter square, which they've asked us to find. So this is the answer. On a number grid, place the quadrant randomly using coordinate from a random number generator. Count the number of each plant species in that quadrant and divide the number by the area of the quadrant. So this is to get the number of each species in 1 meter square of the field. Then we have state what is meant by genome. So we know genome is the total amount of DNA in an organism. So our wording will be like that: all the DNA or genes in an organism. Describe the function between the structure of DNA and RNA. So this is a very common question that structure of DNA, we know DNA has double helix with two strands and RNA is a single strand, so there is no helix. Then DNA has deoxyribose sugar, where RNA has ribose sugar. Then DNA has thymine, but RNA has uracil. So many more like that. DNA is longer, RNA is not that much longer than DNA. So these are the answer. Describe the role of RNA in protein synthesis. So there are two RNAs involved in protein synthesis. That is one mRNA, which is produced in transcription. So what mRNA does is it copies the complementary codes. It copies the codon of the DNA and then it leaves the nucleus through the nuclear pore and joins with the ribosome. So now starts translation where the tRNA molecule also gets involved. So mRNA contains or carries the codon, whereas the tRNA molecule carries the codon, anticodon, and amino acids. So in translation, they bind, the codon binds with the anticodon, and the amino acids are placed together to form a chain, amino acid chain, or a polypeptide. So like this, transcription produces mRNA, part of DNA unwinds, and mRNA copies the code of DNA according to the complementary base pair ruling. Like if there is A in DNA, then there will be U in the mRNA. Then the mRNA moves out of the nucleus into the cytoplasm and binds with ribosomes. tRNA molecules bring amino acids to ribosomes. The codon on the mRNA binds with the anticodon on the tRNA, producing an amino acid chain, or you can say it produces a polypeptide chain. Translation produces an amino acid chain. So that is the whole protein synthesis, but they have asked us to write it in terms or highlighting the roles of the mRNA and tRNA. Then we have the menstrual cycle. The graph shows the changes in levels of hormone released from structure B during one menstrual cycle. So this from this graph alone we can find out what structure B was and what the hormones are. So this is a simple way to find out because at 14 day we know there is a high hormone level of LH. So LH is the middle or the mid hormone release. Before LH, progesterone is released, and after LH or after 14 days, estrogen is released. Sorry, before LH or before 14 days, estrogen is released, and after LH and 14 days, progesterone is released. So we can see immediate before it is the peak and immediate after it is another peak. So this one M is estrogen and N is progesterone. So both of these hormones are released from the ovary. Now let's see, they've asked us explain the importance of the changes in hormone M and N during the menstrual cycle. So the hormone M increases just before 14-day, which is the ovulation. So the hormone M, or estrogen, increases or peaks just before ovulation because the role of hormone M, or estrogen, is to repair the uterus lining and to prepare that uterus lining, or to repair it to the point that a baby or a fetus can be implanted there. It it repairs the uterus lining for implantation of the fetus. Then we have progesterone, which increases after ovulation. So it the function of progesterone is it peaks after ovulation because it has to maintain that uterus lining which was repaired by estrogen. So it maintains the uterus lining, and it also inhibits the production of FSH and LH. So answer is this is estrogen increases and peaks before ovulation, repairs the uterus lining and making it ready for implantation, and also stimulates the release of LH. And estrogen increases after ovulation because it maintains the uterus lining and inhibits the release of FSH and LH. And another thing, when estrogen drops, progesterone drops, then the uterus lining is not maintained, so then menstrual cycle is triggered again or causes menstruation.
Let me guess. You are watching videos, taking notes, maybe even grinding late at night. But the marks, they are just not going up. You are stuck, and it's frustrating. But here is the truth, it's not your fault, it's the method. And that's exactly where Exam Essentials makes the difference. We at Exam Essentials actually offer two very powerful courses to navigate you towards success. The first one is the most popular high impact crash course. If you are just 4 to 6 months away from your exam, this course is going to be a lifesaver for you. And the second course that we at Exam Essentials actually offer is the complete 1 year course. This is basically for the students who want full support from day one to exam day. And the best part, our students' results speak for themselves. Both the courses are actually open ahead of every exam session. No matter whether you are going to give your exam for the May 26, Jan 27, May 25, or October 25, doesn't matter. We always offer these two courses before every upcoming exam session. To join or know more, just check the description box below. You will find our WhatsApp number and Facebook page link right there. Let's turn your effort into results together.
Next up, we have give 3 reasons why scientists use micropropagation to reproduce the transgenic crop plants. So this is used because micropropagation allows to produce identical plants. They can be produced at any time of the year. They can be produced much faster in larger quantities, and also they have a less risk of cross pollination with wild plants. So these are the answer. We have to write any 3 of them. Describe how hydrogen carbonate indicator could be used to investigate the effect of light intensity on carbon dioxide exchanged by chlorella. So we know hydrogen carbonate reacts to the concentration of carbon dioxide in the atmosphere or the air. So if the chlorella has high exchange of carbon dioxide, that means if they're taking high concentration of carbon dioxide into them, then the carbon dioxide concentration will decrease. And at low concentration of carbon dioxide, the hydrogen carbonate gives a purple color, but at high concentration of carbon dioxide, they give a yellow color. So that will indicate the chlorella is not having a good gas exchange of carbon dioxide. They're not intaking that much carbon dioxide, or rather, they're releasing carbon dioxide. And when if they have a fair exchange, such as the amount of carbon dioxide they take in, they release, or the air around them have about 0.04% carbon dioxide, which is the atmospheric concentration of carbon dioxide, then they will give a red color. So these are the indicators, but we have to describe how to do that. So we will move the lamp from different distances from chlorella so that we can change the light intensity. Then place the mass of chlorella in each test tube with the same volume and concentration of indicator, which is the hydrogen carbonate. Then leave it for some times. So if the light intensity at low light intensity, it will turn yellow, because at low light intensity photosynthesis does not happen, or the rate of photosynthesis is slower, which is why they have a higher rate of respiration. They do not take in carbon dioxide, rather they release carbon dioxide, which increases the concentration. And as I've said, at high concentration of carbon dioxide, they turn yellow, and vice versa for red. Or, or you can also write purple. When body becomes dehydrated, the concentration of urine increases. Explain why that occurs, that lead to the production of a concentrated urine. So when our body becomes dehydrated, that means our blood has low content of water. So this particular change or condition is detected by the hypothalamus, and it sends a signal to our pituitary gland to release a hormone which is known as ADH. So ADH increases when the concentration of ADH increases. ADH causes the walls of collecting duct to be more permeable. So if they turn more permeable, then water can happen, or osmosis can occur at a faster rate. So, or happen more often. So water is reabsorbed back into the blood, or water travels by osmosis from the collecting ducts into the blood. Then the concentration of urine increases because there is no low content of water in the urine. So this is the answer. When water content of blood decreases, hypothalamus in the brain detects the change and stimulates pituitary gland to release more ADH. This causes the collecting ducts to become more permeable, and so water is reabsorbed by osmosis. A small volume of concentrated urine is produced. Next off, small intestine have we are asked to describe the adaptive features, or how small intestine is adapted for absorption. So we know small intestine have villus, microvillus, which increases the surface area for diffusion. Then they have thin walls for short diffusion distance, and they have blood capillaries close to the walls to provide a steep concentration gradient. And lastly, they have lacteal, which absorbs lipids. So they're first of all highly folded, contains many villus and microvillus, which increases the surface area for absorption. Then walls are one cell thick or very thin to provide a short diffusion distance. It supplies the blood by dense capillary network to maintain the steep concentration gradient and to absorb glucose and amino acids. And it contains lacteal to absorb lipids. Next off we have cells in human placenta also have microvilli. Describe the role of placenta. So we know placenta have a very important role for pregnant women. So placenta provides the fetus with glucose, oxygen, amino acids, hormones by diffusion, and it removes the carbon dioxide and urea or other waste also by diffusion. Then it also provides transports antibody, and it can also produce the hormone progesterone to maintain the uterus lining. So they maintain a they forms the main surface for transport of oxygen, amino acids, glucose, fatty acids to the fetus from mother and remove the waste such as urea and carbon dioxide from fetus to mother, and these are done by diffusion. It provides antibodies, and they can produce hormones such as progesterone to maintain the uterus lining. Next up, we have explained what is meant by biodiversity. So, biodiversity is the variation in the organism and the number of species along with the number of each species. So, it is considered the variation in the organism shown by the organism and number of different species and each species. State one difference between cloning an organism and genetically modifying an organism. So we know cloning, they're genetically identical to their parent or what from where they have been cloned from. State what is meant by codominant. We know in codominant, both the alleles are dominant, which is why both of them are expressed. So both alleles are expressed in the same phenotype. Describe how saprotrophic fungus such as mold obtains its food. So, saprotrophic means anything that digests their food outside their body then absorbs them. So, this is known as saprotrophic nutrition. So, our fungus here will release the extracellular enzyme on the food, then those extracellular enzyme will digest them outside the body or break down the food outside the body, and they will absorb those digested nutrients through their hyphae. So this is how they obtain their food. Feeds by extracellular digestive enzyme onto the food, then absorbs the digested food molecules by hyphae. They also only feed on the dead or decaying organism. Now we have explained how Explain the conditions needed for seed germination. We know there are main three factors of seed germination. One is temperature, water, and oxygen. Another one could be light. So first, let's come to those three important ones. We need water for to activate the enzymes or for enzyme activity, such as chemical reactions. Then we need an optimum temperature to provide the enzymes their optimum activity so they can carry out their functions more quickly and faster. Then oxygen is needed for aerobic respiration so we can get the energy so they can get the energy, and light is needed to stimulate the growth through the growth hormone. Explain the methods a fish farmer can use to maximize the production of fish. So a fish farmer can use several methods to maximize the production of fish. One of them, or the first of them, is separating the fishes, judging from their age, size, mass. So this is known as intraspecific competition or condition, which means they are of the same species, but they are at a different age, a different size, a different mass. So this reduces the competition between the weak ones and the big ones. So all of them can survive, where mainly fishes with the similar composition, if they're at a similar age and similar mass and similar size, they will be able to survive more because they won't have to compete with someone who is superior to them. So this is known as intraspecific competition. And then we have interspecific competition. So this is the competition between different species. So here there can be different species of fishes. Some of the species can hunt other species, or some of the species may be much stronger, so they are more superior. They eat more food, they leave no food for other species, so we have to separate them by species. Then let's come to food. We have to provide them high content protein food in small amounts but in regular intervals, so that they do not produce more waste or they do not waste the food. So that way we can have them eat all of the food and grow larger. Then we have to clean the water regularly because they do produce waste, so which can accumulate and contaminate the water, and contamination of the water can cause diseases. So the next point would be to vaccinate or to treat the diseased fishes or have them some antibiotics to prevent any diseases that may spread and kill all of the fishes. Then we have to aerate the container or the place where we are there farming the fish to allow aerobic respiration. And finally, we will use selective breeding to maximize the yield. We will have the strong fish bred with each other so they produce a strong offspring. So this will just have a lot of strong fishes around. Describe the role of atria in the body. So we know atria has some functions. One of the or two of them are so no, one of them is to receive blood. So they receive blood twice. One of them is the right atrium that receives blood from vena cava, which is the deoxygenated blood, and one of them, the left atrium, receives the oxygenated blood from pulmonary vein, and lastly, both of this atrium contracts or pushes the blood into the ventricles. Describe how blood in pulmonary artery differs from the blood in aorta. So, pulmonary artery carries the deoxygenated blood and high concentration of carbon dioxide containing blood to the lungs. Meanwhile, aorta carries the oxygenated blood carrying oxygen and carbon dioxide to the body from the left ventricle. So this is the difference, that pulmonary artery or carries more carbon dioxide and less oxygen, or yes. And additionally, pulmonary artery has lower pressure because the right ventricle does not generate as much force as the left ventricle. Then we have the term meant by, what is the term meant by balanced diet? So balanced diet is all of the compositions of food contained in a proper ratio proportion. So all of the a diet consisting of all of the food groups. So there's carbohydrate, fiber, lipid, and other food in a correct proportion. After pollen grain lands on the stigma, a pollen tube grows. Explain the role of pollen tube. So it is fairly simple. Pollen tube grows down the style through the micropyle to the ovule. This allows the pollen nucleus to travel down the style and enter the ovule to join or fuse with the egg cell for fertilization. So they grow down the style, enters the ovary through a micropyle or ovule, then it transfers the pollen nucleus grain to the ovary for the fertilization of egg. A plant root hair cell absorbs water from the soil by osmosis. Explain the structure of the root hair cell that is adapted to absorb water. So we know the root hair cell is very elongated. They have a nose-like structure. So they have an elongated cell or part, so which allows osmosis to happen over a large area. So they're single elongated extension of epidermis cell in the root, which increases the surface area for absorption of water. And then we have the most important one, the role of bacteria or other bacteria involved in nitrogen cycle. So there is only 3 bacteria involved in nitrogen cycle: nitrifying bacteria, nitrogen fixing bacteria, and denitrifying bacteria. So each of them are involved, such as nitrifying bacteria is involved in nitrification. Nitrogen fixing bacteria is involved in nitrogen fixation, and denitrifying bacteria is involved in denitrification. So we also have to know what happens or what each of them do, such as nitrogen fixing bacteria takes nitrogen gas and change it into ammonia into the soil. So they convert nitrogen gas into ammonia. Nitrifying bacteria converts ammonia that is present in soil into nitrite with I, and then converts it further into nitrates with A. So make sure to get the order correct, otherwise you will lose the marks. And lastly, denitrifying bacteria, we can guess from the denitrifying bacteria it will be opposite, that nitrates are converted or turned into nitrogen gas. So these three can form a cycle, so which is why hence the name given nitrogen cycle. Then we are told to describe a procedure that student could use to obtain explants and produce these results. So we don't know the results, but we do know the procedure to produce explant or to obtain explants. So basically we have to describe the micropropagation. So where we cut out part of plant using a sharp knife, which is known as explant. Then we transfer that explant to agar media or agar nutrient containing the sufficient glucose, oxygen, amino acids, and other nutrients which are needed for growth. Then they start growing as we put it in optimum temperature and pH for optimum growth. Then when they grow into shoots and roots, we transfer them to a compost containing fertilizers or nutrients. So this is the answer. We remove explants such as tips of stem and side roots from the same parent using knife. Sterilize the surface of explant using ethanol, followed by rinse with sterile water because to prevent growth of bacteria in the agronutrient. Then grow them in sterile petri dish containing sterile nutrients, nutrient agar, which contains minerals, amino acids, glucose, as I've said. For experiment, change the pH by adding acid and alkali buffer solutions. So we have a buffer so the pH does not change. And count the number of shoots after a certain period. Repeat for reliability. Temperature and density should be controlled. So these points are for the obtaining the results which are not given, but up to before this part up to this is the most common asked question known as micropropagation. So this is the answer. And then we have a transverse section of leaf. We can see that. Explain how layer A is adapted for its role. So A is the upper epidermis which with a cuticle layer. So it has a waxy layer to prevent evaporation of water, and also it allows the sunlight to enter the palisade layer. So it is thin and transparent to allow light and has a waxy cuticle layer to prevent water loss, and it also can protect against infections. Explain how layer B and C are adapted for photosynthesis and gas exchange. So let's look at B and C. B is the palisade mesophyll layer and C is the spongy mesophyll layer. So palisade mesophyll layer, they are tightly packed with each other containing a lot of chlorophyll. So they are mainly adapted for photosynthesis. And then we have the spongy mesophyll layers which have gaps in between them, and they also have chlorophyll. So they do carry out some photosynthesis, but they also carry out gas exchange because they have the gaps within them, so diffusion can happen. So this is the answer. So the palisade mesophyll layer B, mainly contains many chloroplasts tightly packed for maximum absorption of light. And the spongy mesophyll layers are loosely packed with cells with internal space, so allows gas exchange such as diffusion of oxygen out and carbon dioxide in. Explain how layer D is able to regulate gas exchange. So layer D was the last layer which are the guard cells. So they're the guard cells which control the gas exchange enter of gases. So mainly they control the gas exchange by opening or closing the stomata. So for tumors we'll write that D has many Stomata with guard cells which can open and close the stomata and lets the carbon dioxide in during the day. Now we have to describe the stages how to clone a male dog. So in order to clone we need an adult cell and a enucleated egg cell. So we will take the nucleus from the adult cell and join or transfer it to the enucleated egg cell, join them by electric shock, and then mitosis will occur, forming an embryo. Then that embryo will be transferred to the uterus of a surrogate mother. Next off, we have to describe the types of pathogen. Human populations are at a risk of infectious disease. Describe the types of pathogen, refer to a disease that each type of pathogen can cause. So this is the only time this type of question came, and this is the unique question that most of the students weren't able to answer because we have never seen anything like this before. So we have to write down different types of pathogens such as virus, bacteria, fungus, protozoes, and their diseases. So let's look at that. As I've said, there are pathogens such as virus, bacteria, fungi, and protozoes. Virus, we have to describe their properties or their features. They are microscopic single-celled organism with no true nucleus, or you can say they are prokaryotic cells. They cause tuberculosis. So this is the disease they cause. Not all of them, but some of them. So one of the disease they cause. Then viruses are parasitic particles. They only have protein code and genetic material such as DNA or RNA. One of the diseases caused by viruses is AIDS. Then we have fungi. They are eukaryotic organisms with cell walls made of chitin. So this is the unique feature of them. And one of the diseases that is caused by fungus is athlete's foot. And then we have protozoans. They are many single-celled organisms with nucleus. And one of the diseases they cause is malaria. So this is a very unique question. We do not have such names of diseases caused by each pathogen. So better you have all of it memorized so you have the examples of diseases ready if they ask you the same question. So next we have one of the most important roles of hormones. So in this table we have all of the hormones and their locations where it was released and their functions. So let's come from FSH. FSH, sorry, FSH is released from pituitary gland and its function is to stimulate the growth of follicles and to stimulate the release of estrogen. Next off we have LH, which is also released from the pituitary gland. It stimulates ovulation and the development of corpus luteum. Estrogen, it is released from ovaries. It repairs the uterus lining and stimulates the release of LH. And lastly, progesterone, which is also released from ovaries. It maintains the uterus lining and inhibits the release of LH. Then we have the term meant by menstrual cycle. So this is the uh, monthly blood loss that happens due to the breakdown of the uterus lining. So It is the monthly blood loss due to the breakdown of uterus lining. Next, we have a fermenter, which is used to grow organisms. So, describe the function of paddles in the fermenter. So, here, this is the paddles. So, they are used to mix the contents together, to mix the microorganisms, the nutrients together, and to evenly distribute the oxygen and temperature for aerobic respiration and optimum enzyme activity. So evenly distributes the heat and oxygen, and to mix the food microorganisms with food substrates or nutrients. Then we are asked to explain why the fermenter has a filtered air supply. So it's for 2 marks. So the key word is filtered and air supply. Filtered because we do not want other microorganisms to enter our fermenter and start growing, starting a competition with our desired microorganism. And air supply is to supply with oxygen for aerobic respiration. So this filter removes the other microorganisms to prevent competition. And they've asked us to explain what is meant by the term sustainable. So sustainable is anything that does not run out. So it is maintained and does not run out. Describe the role of insulin. So insulin is a it particularly changes or converts glucose into glycogen, lowering the blood glucose level. So it lowers the blood glucose level by stimulating liver cells to take up glucose from bloodstream and convert it into insoluble glycogen. Then we have describe how bacteria are genetically modified to produce human insulin. So basically we have to describe how a bacteria is genetically modified. So for modification, we know we are using two specific enzymes. One is restriction enzyme to cut up the gene that is used for insulin production, and ligase, which is used to join that cut part of gene to the plasmid of bacteria. So we will write that gene is cut for insulin is cut using restriction enzyme, and plasmid for bacteria is cut using the same restriction enzyme, and both are joined together by the ligase so that they form a recombinant DNA. An industrial fermenter is used to grow the genetically modified bacteria. Explain why fermenter is cleaned using steam before genetically modified bacteria are added. So it is cleaned with steam. So steam has a very high temperature, so it is used to sterilize the fermenter by killing other microorganisms which may allow competition or increase the competition. So to prevent the chemical contamination, so if there are other chemicals build up, we are removing those too, and to remove pathogens for reducing competition and to get the conditions aseptic. Then we have describe how you would test a leaf that contains starch. So for we are basically doing starch test, which is we are going to use iodine solution on the leaf. First we have to discharge the leaf. So we will boil it in the boiling tube, then transfer it in hot ethanol, then back into the boiling tube to remove the ethanol. All this to remove the chlorophyll, and then place it on a white tile, add a few drops of iodine solution. Positive test will show blue-black color. So The result shows many alveoli increase the surface area, which increase the rate of diffusion. Explain three other ways that alveoli is adapted to maximize gas exchange. So there are many functions. Alveoli is has thin walls, has a good supply of blood, it is moist, and it other things are not. There are these three things, that it is one cell thick to provide short diffusion distance, then it has a steep concentration gradient which is maintained by dense capillary network, and the lining is moist so the gases can dissolve in.